I'll do a bit of an introduction before each of the speakers. So first, we will have Rafał Kamiński. You will be able to share your screen in a moment. Rafał has international experience in discovery and development of innovative small molecule drugs. After obtaining medical doctorate in pharmacology in Medical University of Lublin, he completed his postdoctoral training in the Netherlands and USA, and as well obtained the diploma in pharmaceutical medicine. He joined pharmaceutical industry more than 13 years ago and worked for UCB Pharma and Roche, and now he's with OncoArendi Therapeutics. So Rafał, thank you for joining, and we're looking forward to hearing about your industrial experience so far.
Thank you very much for your kind introduction, and thank you for the invitation for today's event. I'm really excited to take part in this session. Do you see my slides? Is it everything okay? Yeah. Okay. Right. So we'll be talking today about success in drug discovery, and this success is plagued with failures. So we'll be trying to think how to minimize, the risk of attrition and increase the success. And when we talk about success, this is how most people think about success, including success in drug discovery and development. You have a great idea, you launch a program, and then you're on the market. It's pretty much like a straight line, it seems. But unfortunately, it looks more like this. It's much more, tangled and convoluted process, and sometimes we get lost in this process. So there are strategies that we will be talking about today that try to make this line as straight as possible and reach the end goal, meaning bringing drugs to patients as quickly as possible. So what is the attrition and in R&D, in drug discovery, and what's the scale of this problem? When you look at this graph, it illustrates, it's based on a number of development programs, discovery and development programs, actually I think more than 1,000. And based on this data, you can analyze what's the probability of success, starting from preclinical research to launching a drug on the market, and that's the first tiny bar on the left, and you see that the cumulative probability of success from that stage to market is about 5%. Then when you move to phase 1, these are studies in healthy volunteers. It's inches up, but still it's well beyond 10% on average. So only 1 of 10 compounds or products at this stage reach the market. Then it starts to increase and the process gets to risk. So phase 2 would be studies in patients, actually smaller studies in patients, proof of concept studies and confirmatory studies. Then the probability increases. Only when you get to so-called phase 3 studies, these are large studies involving hundreds, sometimes even thousands of patients. This is a large population. Then the probability increases further. And then once you completed the studies, you have to file these regulatory dossiers, applications for new drug application or biologic license application. And even there's no guarantee that the regulators would approve a product for the market. So this is rather risky endeavor, and it fails for most of the time. So it's so important to make it efficient. I gave you on the previous slide some average numbers. Here you see these numbers broken down by indications and some sort of large categories of indications. And you can already appreciate that some type of therapeutic areas or some diseases have higher probability of success. For example, infectious diseases, autoimmune diseases. But on the far right, you have the smallest probability of success, and that's actually in oncology. And as we all know, this is probably the biggest area of unmet need today, yet the least successful. So this also shows you, as consistent with previous slide, that there is a big difference between phase 2 and phase 3 studies in terms of success. There is also, we can break this down, these numbers into specific therapeutic modalities. So we think about sort of pills and injections, but there is actually a whole host of different type of therapeutic modalities. Actually, this list is ever growing, CAR T cells and gene therapies. I think we have a talk later in the session about gene therapies. So when you look from this angle, indeed, the chances are also not even for different types of modalities. And when you look to the bottom, where still majority of programs go on, so small molecule drugs, this is about 7.5%, whereas gene therapies and especially these RNA interference therapies and CAR T therapies, as they are much more targeted, they have a higher probability of success. And on this graph, you have this likelihood assessed from phase 1 to approval. So what are the reasons for this attrition in research and development, in drug discovery and development? So the first reason is what we could call scientific. I think most of what we do is science related, but there'll be a specific category of so-called scientific reasons. So here, in the preclinical space, mainly, there are problems with achieving proof of concept, showing convincing efficacy, and most importantly, also safety in preclinical species. Then once the program moves to the clinic, then the issue becomes clinical efficacy and safety, particularly in the beginning of these programs. And as I will show you later, the main reason for that is the sort of weakness of the link between disease and the target. So the documentation and convincing evidence about pathological role of your target in disease. And there are also issues related to bioavailability and pharmacokinetics of drugs or different drug substances that are being developed. And this would be more related to technical reasons. So, for example, things as trivial as formulation of drugs could make a difference and could be a reason for failure. There are different manufacturing issues that can also cause problems. And intellectual property and legal issues can also actually stop development programs. There is a raft of operational issues, especially at clinical stage. These are typically big projects involving a large number of people globally. So they need to be very well orchestrated. And sometimes we embark on diseases for which there is no treatments yet. Where there's high unmet need, but no one has done it before, and sometimes there's no clear regulatory path. There may be different registration issues, data quality issues, and something that most studies struggle with currently is patient recruitment, sometimes patient compliance, so patients stay on a trial until the end. Then there are commercial reasons. Obviously, this is commercial activity. So there are reasons such as, for example, cost of goods. Some drugs or some types of therapeutic modalities are so expensive to make that actually it's very difficult to make a profit on them in the future. There are oftentimes, especially in large pharma organizations, strategic decisions, portfolio changes, prioritizations, and so forth, and programs are stopped. Sometimes there is a transformative treatment that comes on market and makes the commercial value of a given program basically useless or minimal. So the competition is also very, very important factor here. Now, when we look at some more granularity, main reasons for late stage development, so failure in late stage development, phase 2, phase 3, is actually efficacy. This red, glaring red color, more than 50% studies fail for efficacy, and then safety and strategy are the main reasons at that stage, so late stage development. When you actually look at earlier stages of development, and this was only possible to analyze in one organization, in this case AstraZeneca, they have very rich pipeline of drugs, and these projects can be objectively compared. It's very difficult to get these data across industry. So you see that this interesting shift from preclinical stages through phase 1. This is where mainly safety aspects and unexpected safety aspects, tend to stop programs. Once these drugs get in, or drug candidates get into late stage development, phase 2 proof of concept and phase 2 confirmatory studies, then the efficacy comes in, and that just shows that typically drugs work much better in experimental models than in humans. And that's a big surprise. Everybody hopes it will be confirmed what we see in the lab, but for most of the time, unfortunately, it doesn't happen in the clinic. So this AstraZeneca study looked really in detail what are the reasons for lack of clinical efficacy. And as I already mentioned briefly, the key reason is the linkage to the disease, the target linkage to the disease. Sometimes, there are no confirmation of target tissue exposure, especially in CNS diseases. This is very difficult to measure, concentration of the drug or target engagement in CNS compartment. Sometimes in oncology, that's also very difficult to do. Then sometimes there could be a mismatch between the target population and the indications. So diseases, common diseases are very heterogeneous. They have different genetic and molecular reasons, causes, yet the symptoms may be similar, but the biology is quite different. And that mismatch can happen. And sometimes there is some kind of wishful thinking in the industry, and we'd like, obviously, our projects to succeed and go from stage to stage, and we make not robust enough decisions and go forward with the data that may not be really convincing. But the key reason is this target validation, and that's why I think there are strategies focusing on addressing that key issues. And one, so how we actually reduce this attrition and what we can do, and does it actually work? So first, the target validation, as I mentioned, is the key problem, and one way to address this problem is actually stronger reliance on genetics and genetic data, and here you can have examples of types of genetic data that are available that can substantiate the rationale behind therapeutic target. Historically, these were GWAS studies, so genome-wide association studies, where you can associate a genetic loci in a particular gene with a disease or risk for disease. Nowadays, I think it's more next generation sequencing data, so exome sequencing or whole genome sequencing, and that opened up a number of monogenic diseases where there are causal genes that can be targeted, and that gets us very close to the disease causing problem and increases the chance of success. And then also transcriptomic data, gene expression data can inform us about important pathways that are driving diseases, and intervention at that level tend to have higher success rate. So here, when you look at what different it makes, so these so-called genetically validated targets, so those where there is genetic evidence behind, they have much higher probability of success, actually twice higher than targets without genetic validation. That what's illustrated in this green bar, and this is cumulative from phase 1 to approval. When you look at particular phases, it's still inches up versus these non-genetically validated targets. So I think now across the industry, we'd like to make sure that the targets that we work on have this genetic validation. The other area where it can make a big impact, actually as much as threefold increase the chance of success in case of the cumulative probability from phase 1 to approval, is the so-called biomarkers and stratification biomarkers. So you can select very precisely a patient population based on genetic and molecular markers. So not everybody with the same symptom, as I said, would have the same reason for the symptom. But if you stratify based on molecular and genetic biomarkers, you increase the chance threefold. So actually biomarkers is a big field, and all of them that I'll explain very briefly in a minute should be applied to every drug discovery and development program, starting from drug exposure and measuring the concentration in blood and in target tissue, and making sure it's high enough but also safe enough. The target engagement, target occupancy. So you have to demonstrate that your therapeutic modality does interact with the target. Not only it binds, but it also exerts some kind of functional effects, and that's called target engagement. Then obviously we have to be very careful and look at safety aspects, particularly for small molecule drugs. They have oftentimes this liability to have untoward effect on cardiovascular system that can be de-risked in preclinical species. Study endpoints are also important, and here it's critical to have objective measures, not the kind of subjective clinical scales, but really use imaging or functional tests, biochemical tests, really to measure objectively what the drug does to the disease. And then patient selection, I've already alluded to this. Something that I'm just flashing out as kind of novelty, but very rapidly growing, and I'm sure Google is and other companies are looking into that very closely. They actually make big business on this. So a number of important data regarding health and well-being can be gathered with so-called digital biomarkers, and they are objective and quantifiable and can be associated with meaningful disease outcomes or symptoms, and also can be integrated with a host of genetic molecular data. So they just paint much broader picture than just these traditional clinical scales. So, I mentioned to you a few ideas how to improve this attrition in R&D, but does it actually work? Well, it seems it does, and especially since the time where the industry as such has critically looked at these reasons for attrition. So that would be the first decade of the 21st century. And when these strategies were put in place, we now see that there is some kind of gradual increase in success rate across indications. I couldn't find more recent data than that. Hopefully it will be available soon. So we'll see if this is a true trend or just a blip, but it seems to be working. And the next sort of reason that illustrates that it seems to be working is this. So when you look at 20 years back of FDA approvals of new drugs, it seems like the last 3 years were really record-breaking, and there is the biggest number of new therapeutic modalities drugs being brought to the market. So I think the industry has learned the lesson, and we as such became more efficient in bringing new drugs to patients. So very briefly, just to summarize, what are the key elements of these strategies that are meant to improve success. So first is biological targets. So really making sure and taking time in the discovery phase to substantiate targets that we work on with human genetics and biomarkers to make as strong as possible link to the disease. Then, when we work on a particular organ, it's important to confirm that in this organ, in this target organ, we engage and occupy our target and also have sufficient exposure. I mentioned to you about safety. This can be, as I said, the risk much earlier than in the clinical stage. So we don't put patients at risk, and studies in the end don't fail because of surprising risk or untoward issues. But also, I think this has to be looked at as a balance between benefit and risk. In some severe diseases, patients are willing to take a bit more risk, especially those that are life-threatening diseases. Then patient population, again, critically important, and this patient selection element. So not enrolling everybody into studies, clinical studies, but pre-selected patients based on molecular and genetic evidence would really make a difference. Then clinical studies should use these robust and objective efficacy readouts, and also it's critically important not to introduce too many endpoints, because that creates noise and reduces statistical power. So really, we need to be selective about these clinical endpoints in our studies. And finally, the commercial potential. It's really important to bring a value proposition that it differentiates from the current or sometimes even future standard of care. So we need to envisage how the field, how the treatment standard of care will evolve, and how we will fit into that years ahead once the studies are finished. So at the time of launch, not at the time of beginning of the project. And then it's good to do these in alignment with payers who reimburse the drug costs, healthcare providers, and most importantly with patients themselves. So this is in a nutshell, and within 20 minutes I was able just to signal to you some of these elements, but thank you for your attention. I'll be happy to discuss and take questions.
Thank you very much, Rafał, for the really interesting talk. And now that there will be time for questions, so please either raise your hand or write them in the chat. Oh, I see now first question coming in the chat, so I'll read it. Great talk from Tomek Włodarski. Is it possible that the increase in FDA-approved drug is just due to more money put into the research and the fact that there are many more companies right now than 20 years ago, rather than we are doing something better?
Yeah, thanks for a great question. Actually, I think it's not really correct a sort of view on why we have more drugs being approved. I think genuinely these strategies work, and there is objective increase in productivity and success. So it's not just a function of numbers, but these are proportional data. So we're becoming more efficacious. Yes, indeed, there is more money, but actually the sustainability of the industry as such becomes questionable. And now the latest data show that it takes about $2 billion of dollars to bring a drug to the market. Obviously, that's not a cost of a single development program, but cumulative R&D costs divided by a number of drugs that the industry actually brought to the market, and this is a staggering figure. So it's just becoming more expensive. It's not that more money is available, it's more expensive. So that's why it needs to be so efficient. So I would still maintain my view that that's because of better designed programs, better designed drug discovery and development programs that makes it successful. And also maybe one more comment that perhaps didn't transpire when I was mentioning about these patient populations. Yes, now the industry likes to go more into personalized care and subpopulations of patients within one indications. The kind of blockbuster model, one size fits all, doesn't really work. So that translates into more programs, more drugs. But yeah, they are, I think, brought up more efficiently to the market.
Great, thanks a lot for the answer. There's another question. How do you strategically choose which clinical endpoints to focus on? This is a question coming from Iga.
Right, so that's very much dependent on the indication and dependent on the stage of development. So in early stages, well, phase 1, phase 2, these are so-called exploratory stages, so you can be a bit more flexible with your endpoints, and there you focus on these elements, as I mentioned, target engagement, you know, target validation. But when you get to the later stages, then you have to be more restrictive and even more compliant, so to speak, with guidelines, regulatory guidelines. And for most indications, the regulators would actually tell you what endpoints to use, and based on which endpoints they would approve a drug for market. So there's not so much flexibility there, what you can use. But again, in short, this is very much dependent on the phase and on the type of indication you're working on.
Great, thanks a lot for these explanations. We have still two more minutes, or 1 more minute for a question. I actually would like to ask one myself regarding the digital biomarkers. What do you think is the future in your field of the digital biomarkers?
Yeah, so as I mentioned, this is growing rapidly, and nowadays, I think at least big pharma companies, pretty much every company in late stage programs, they would have digital biomarkers. So these wearable devices that read biometric data, or sometimes these portable devices that can be used by patients to enter you know, their well-being and the symptoms and so on. So collectively that creates a large amount of data. So I think this is really future, and even now becoming more important in the COVID times, because now the industry also has to adapt to the situation, the pandemic situation, and more studies are done remotely, involving fewer and fewer visits of patients in clinical centers. So there's ever growing reliance on these types of devices, and they're pretty accurate now and approved actually for clinical use.
Great, thanks a lot for the really interesting talk and the answers now. So